Okay, so this is the second part of our discussion of genome complexity, where we're looking at the big pictures. The last one we introduced the ideas of when you stain up your chromosomes, you often see that there are two basic types of regions within a chromosome. We call those euchromatin, those are the lightly staining ones, and heterochromatin, which is the more darkly staining ones. But how do, that's just individual chromosomes. What about the whole genome? What do we learn? Well, it turned out there was a technique that could be used to determine the amount of DNA that was actually in the species, right? Once we understood that we were interested in DNA, that's how much was there. And this was generally scaled down to the haploid genome of a species or whatever. And so people started looking at all sorts of things once we could figure out that, look at the DNA and saying, okay, this species has this much, this has this much, what's going on? And this is sort of a representation of that. It's not perfect, but it's a good representation of the haploid genome. Notice these are going to be log standards down here. So it's an order of magnitude change from one to the other as far as complexity, right? And if we plot a line about genome size, it looks something like this. Way down here, fairly small as you would expect, are viruses, tiny, very tiny as far as their amount of genetic material they would have. Bacteria can have more, etc. And then we start moving up the sort of the evolutionary scale. We see fungi and algae will have more because they've got more components to them as far as their structure and function, the way they reproduce. And then we have things like flatworms and insects, and everywhere we cross the line right here is about the average. That's what this line, this blue line in the middle, is the average genome size. Okay, it's not, you don't need to memorize the genome sizes. This is just to give you an image. And then we move up to echinoderms, okay, starfishes, and they're somewhere up in this region. So obviously, as we move up the evolutionary ladder, right, we see that we have an increase, as you would expect. Things are getting more complex. Okay, fish are more complex in general than insects. They have more need for genetic material, more things that are going to occur. Right. Certainly, amphibians have a huge range. Amphibians come out of the water, right? At least as adults. Then they go back, so they're in and out of water. They do all these things. Birds are even more on average, because remember the average is here. This green bar is the amount of variation that you would see, but these we're talking about right here. So on average, the birds have a larger genome than say an amphibian. Reptiles are slightly larger again, and then mammals, one of the more evolved systems, have this. Okay, so we see a nice little, relatively strong trend as we go up what we would consider the evolutionary pathway to more complexity of the organisms. Now, what do you think has the largest amount on average? Well, it turns out to be flowering plants. And people go, oh, wait, flowering plants, wait, mammals, they breathe air, they reproduce, they produce milk, they have mobility, they do all these things. Plants just kind of sit there. Why would they have more DNA? And then look at this huge amount of variance here. And then they started looking back and saying, well, look at the variance here, And then in reality, insects here, I mean, they have respiratory systems, spiracles and things. They have a circulatory system, neural systems. They have muscles. They fly for many cases. Why? They're not that much less complex than a fish. Why would there be orders of magnitude more DNA in a fish than you actually have in an insect? People really got confused about this. And what's all this variance out here? Why do some amphibians need, fairly small amount, and some need, 100 times more DNA? What is that? What's going on there? That became the question once these data became available, and it became known as the C-value paradox, right? And the paradox is fairly simple. There appears to be more DNA than we actually need as we go up the evolutionary line. Well, what is all this DNA? I mean, these things are, an amphibian, yes, it does some things that a reptile doesn't do, but they both locomote, they both, do various things, they both produce eggs, they go to reproduction. What in the world is so much more DNA there? Okay, and certainly true when you get to mammals as well. And then, even when you look very closely, like we did at some of the amphibians and things, fairly closely related things often have massive amounts of DNA differences that they would have in their content. And that makes no sense. I mean, you can't imagine that two species that, of lizards or something, so we're moving out of the amphibians, but some lizards and reptiles, that one would need 10 times more DNA than the other to do the very same things. These are just the paradoxes. So what did people think? Well, they said, well, one of the things we think about is clearly as we go through evolutionary time, and if you've ever looked at or taken a course in development, a traditional course where you actually look at the development of organisms, of vertebrates, you see that many things happen as we go through these fetal stages, right? We develop these gill slit looking structures, which are clearly a leftover from the time that we were fishes and our ancestors were fishes, and they don't form gills, but the gill slits and part of your chin and things are clearly uh, evolved, and they're still kind of there in that sense. So but we don't use them. We don't carry gills around, but maybe we carry all the DNA around. So literally, people figured that, a lot of this stuff, DNA must just be junk. We just can't get rid of it. We didn't have an effective way. It's kind of like, if you've ever been in your grandparents' attic and looked around, everything they ever had and used got put up there somewhere, even though it may not, some people don't even know this is called a typewriter anymore, right? Before we had computers, that's what you used. And so stuff is just there, in some form. And the same with your genome. That's what people thought. They thought, well, it must be that our genome, we evolve these new traits or characteristics or new phenotypic elements, but we just don't get rid of the old stuff. Okay. So it became a really big question. It became a big question: is it really junk? Is that what it is? Okay. Um, and one person's junk is another person's prize, right? This is a rat rod down here. It looks like a piece of junk to most of us who have fancy, modern cars. But if you happen to be an enthusiast for hot rods, it's a very valuable piece of equipment. This ugly thing right here probably costs more than your car, unless you're driving a Range Rover or something around. This costs more than a basic new car. I'll guarantee you. Despite what it looks like. So is it junk or not? Right. Same thing with your genome. Well, a technique came along that was associated with this ability to measure the genome. It's a fairly simple technique. So the idea is this: you've got DNA double-stranded, right? Two bonds between A and T, three bonds between G and C. If you heat DNA up, it will become single-stranded. It will just denature, as it's called, right? And then all you got to do is let it sit there and watch it. And you can do this. You have to do it with this, spectrophotometer, but because you can tell the difference between single-stranded and double-stranded by the readings. But you heat it up, and you let it cool off, and you watch it come back together, and you see how long it takes to come back together. And what happens is that regions, and you usually chop the DNA up some. You don't leave a whole chromosome. That's just techniques. Um, Areas where it's a fairly simple system will come back pretty quickly, right? So you may have repeats or that sort of stuff going on. Areas where you've got genes, well, they have to piece. They're not just kind of sitting like this and fold back. They're over here like this. They have to come back and they have to really work to get aligned, and so it takes a lot longer. So areas that, and so it was a technique, okay? And people did it with different species and throwing them together and seeing how similar their DNAs were and all sorts of things. But what they got from this was what's called a COT curve, COT, concentration over time. The time being how long it takes for this to come back. The concentration being how much of your DNA has gone from single-stranded back to double-stranded. Okay, and when they did a COT curve for mammals, you see this. Okay, you see that it has these plateaus early on. This is very small DNA down here that comes together very quickly. Then there's a kind of an intermediate plateau where you've got some larger DNA that takes a while to come back. And then finally, you've got a final plateau that's out here. Okay? And just if we were to look at, say, a bacterial genome, uh, it would just kind of come along here and so this is 100% denatured. Everything's denatured. When you get down here, it's 100% back together. That's what this does. A bacterial genome would just kind of go straight to its size. And wherever it ends up here is an estimate of size as well. There's really no bumps or plateaus in bacterial. It just kind of, it's all genes. It just kind of goes like this, okay? But we have, in ourselves, we have these in other mammals and in other organisms as well. We have these three plateaus. This first plateau is called the HR, which stands for um, highly repetitive. This large area in the middle is called moderately repetitive. And then finally, this is called unique out here. So HR is highly repetitive. Okay, that's where we'll talk about what that means in just a moment. your MR is moderately repetitive, and then we're going to call this area unique. And these are big categories, okay? Again, just like heterochromatin and euchromatin are kind of big categories, and not all euchromatin is can be heterochromatin on that. These are just big general things that are noticed, but certainly these plateaus were noticed, and they turns out they do represent some of the complexity that we see in our genome. All right, so let's take a look at those. What is highly repetitive? Well, this is the least complex. This is the stuff that comes back very, very quickly. So it's usually areas where you've got repeats or something else. Okay, not a lot of uniqueness to that DNA. The moderately repetitive takes a little longer, and it's usually bigger fragments that take a little longer to come back. And then finally, we have our unique DNA, the most complex. Okay, so we're going to work from least complex to most complex. Least complex is highly repetitive. These are clearly non-coding. It's not big enough to be a gene. Stretches of DNA to be genes usually are somewhere at least 1,200 bases, but often the average is over 5,000 base pairs, and they can go into the hundred thousands of base pairs to make a gene. And these we're talking about just very small areas that consist together. They rapidly reassociate, which means they must have simple sequence. And in general, they're a few base pairs long, 5, 6. And what I'm talking about are repeats of this, not not the fact that that it's only 5 base pairs of a fragment to come back. But the similarity only goes for a couple of bases in length. Because of that curve, and you can figure out kind of how much DNA they represent somewhere between 5 and 10% of our genome. And they tend to be clustered around the centromeres and telomeres, as you expect. Those are constitutive heterochromatin in the areas where you don't have a lot of coding going on, but they can be found anywhere. And in fact, they are scattered throughout chromosomes. So some of these deserts, as I called them earlier, or areas where you don't see a lot of genes, they're occupied, a lot of that by highly repetitive. They probably play a role in chromosome morphology in helping to hold the nucleosomes together, or helping to other forms. They're likely to be involved in numbers of other things, locations where proteins are sitting down and knowing to go, etc. One of them we know that they do are these microsatellites that we've talked about before. These are dinucleotide, AT. Well, microsatellite regions will come back together pretty quickly, because there's, just all you got to do is hit an A and a complementary T, and it doesn't really matter if you hit exactly the right one, they'll come back quickly. So we know that they're microsatellite regions, but there are also lots of other things that relate to that are repetitive. Okay. Now the moderately repetitive, again, these are generally non-coding because they're too small. So by that I mean they don't make protein. They're not what's going to make phenotype per se, right? They're not a classic gene, but very often they are stretches that are transcribed. We talked back in the last lecture about some of these nuclear RNAs that potentially shut down euchromatin to make it into heterochromatin. Well, that could be some of this area. So we know that they can be transcribed, but they don't end up making protein. Okay, they're fairly small. There are groups of them, 150 to 500 base pairs, maybe slightly longer than that, and they're repeated. They're all over the genome. Okay, they can be found in large numbers throughout the genome. When I say they, I mean a particular sequence, AT, GG, 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 CT, CG, whatever, 150 bases long. That whole thing is found again and again and again, okay, scattered throughout. They also represent somewhere, and the estimates vary, somewhere between 5 and 10% of the genome. But we do know they can be highly repeated. Some of them can be repeated hundreds to many hundreds of thousands of times in our genome, just depending on what they are. Right. So those are our moderate repetitive. Our unique DNA, these are the ones that are coding for sure. Okay. They're anywhere from a little over 1,000 base pairs in length to hundreds of thousand base pairs in length. And we know now that there's approximately 20,000 genes in humans, maybe 19,000, maybe 21,000. It varies back and forth about what we're actually calling a gene, but somewhere around that level. Okay. Taking that many genes and kind of figuring out the average size, they represent somewhere between 1 and 5% of the total nucleotides in In humans, it's been estimated that they're down around 1.5. In other species, they're up closer to 5%. Just depending on the complexity. Right now, based on genome sequencing, we're saying somewhere around 1.5%. So that's not very much, okay? These are the coding regions. They produce our thing we call genotype, right? The unique areas of these 20,000 genes are what make us us. They're the things that actually get produced and finally ultimately make phenotype because they are our genotype. Now, what's interesting here is you do all the math and you say, okay, let's just maximize it here. Let's say that highly repetitive is 10%, a moderate repetitive is 10%, may or may not be that high, the 1.5%, somewhere under 22% of our genome can we account for by those things. I mean, so what's the other 75%? Well, that's what some of these things are down here that are been hanging out. Pseudogenes are genes that pieces of DNA that were genes that have replicated somehow, maybe been part of duplication events by chromosomes and no longer function. We have a lot of those. Are there genes that we were using at one time in our evolutionary past, like gills, potentially, that we might be carrying pieces of? Yeah. There's non-coding RNA. There's lots of regulatory structures. It turns out, as we'll see when we start talking about what a gene actually is, that you got to regulate it. And that takes up a lot of the space that we have. There are these things called long interdispersed repeats, short interdispersed, and there are these things called introns that we'll talk about as we talk about genes, introns and exons, pieces of our chromosomes that are genes that we don't actually use to make protein but get transcribed. And then we still don't know what a lot of it is. I mean, there are people out there doing lots of work with bioinformatics, just trying to figure out for various species that they're working on what the heck all this other stuff is. Is it functional? Is it just stuff that's sort of our junk? But a lot of it's certainly not junk. It's required to make the system work. Okay? And just to kind of give you an idea here of what we're talking about, here's human chromosome number 1. Okay? It's about 85 millimeters long if you were to take it compressed up. Okay? And that's taking the whole DNA, not compressed up as a chromosome per se as a metaphase, but in fact letting it stretch out and be beads on a string. There's 250 million base pairs, 12 million known variants. So that means there's 12 million known right now places where we've got alleles, if you will. Okay, we've got mutations that are there. That's not particularly important right now. We'll talk about those later. What's important for this discussion is there's a little over 2,000 genes that have been found on chromosome 1 at this point. Okay, there's about half a many of those that are pseudogenes that are clearly have genetic, look like they were coding at one time but no longer are, that are carried around at various places. There's non-coding RNAs that are very long, so these things get transcribed, but they don't produce the message isn't typical message that you use for translation. There's almost 500 of them that are short. There's ones that remain that are microRNAs that are used for regulation. There's ribosomal RNAs that we'll talk about later when we build a ribosome. There's small nuclear RNAs. There's all sorts of RNAs that are found there. And when I say RNAs, it's obviously not RNA in the middle of our chromosome. That would be, that's not what we have here. What we're talking about is that piece of DNA right there actually gets transcribed into a piece of RNA, but that RNA is not part of a gene. That's not what we're making. We're making some other piece of RNA that could be functional. Okay? So my point is, if you take chromosome 1, our largest pair of chromosomes that we have, we've got a couple thousand known genes on there, and we've got lots of other stuff on there. And these you could consider genes because they're transcribed, but they don't get translated. Okay? And they're certainly found all along, scattered throughout these regions. So what do we know? Now we know a chromosome. We know that it's got dense areas, and you can kind of see it here, right? You've got a dense area of, this dense area here is the number of genes found there, not the compression. So a lot of genes for a while, then not many genes, a bunch of genes, not many genes, a big section, a lot of genes, not a lot of genes, etc. Okay? And then there's all sorts of other stuff that's found in these regions as well that make it up. Okay? All right. The next lectures, we're going to actually move and we're going to go in here. We're going to find a gene, one of these coding genes in particular, and we're going to talk about what exactly it is. What does it look like? Obviously it's ATGC all strung out in some way, but are there units of that we can talk about.